So what we're going to do now is we're going to go rewind a little bit because we did the first part of the reflex lab. That was part one, the one that was a hypothesis with the Gendrasic's maneuver and all that. But the second part is just looking at a variety of typical reflexes, that you should be familiar with. And I will kind of go over them a little bit with you. And then we're also going to do a little bit of get all going into breakout rooms. And there's part of it that makes sense to do together. So let's take a look at how this is going to work. All right, so we go. So reflex lab. Just so you recall, reflex action. First, we had this facilitation of muscle stress reflex. This is the stuff we've already done. Hopefully, in fact, just as to get everybody back in the kind of mode of the reflex lab, what was our basic conclusion from doing part one? What did we learn? Anybody?
There was a higher distance when they did like whatever maneuver.
Yeah,
Gendrasics.
Yeah, or kind of, greater mag. So the magnitude of the reflex increased. Remember, so hopefully that should all be relatively recent and clear in your minds. So the next parts were just demonstrations of various reflexes. Plantar reflex. If you remember, plantar just means the bottom of your foot. If you've been in anatomy, it's just a regional term for the sole of your foot. This is a classic thing you do on a newborn. It's part of the reflexes you do when a baby is just born to get a readout on the health of the nervous system. And it's basically you kind of drag kind of a sharp object along the sole of the foot. If you're an adult, the classic response is to curl your toes. If you are a baby, or less than a year old, I should say, the classic response is called Babinski's sign, where your toes actually go outward. So if you're a baby, and they think it's because the motor pathways are not fully myelinated, but you actually have what's normal for a newborn is not the same as what's normal for an adult. If you have Babinski's sign as an adult, that means there's damage to your motor pathways. So this is a typical thing. You just kind of pull this little thing under, and we have videos. I'll show you some videos of this in a few moments. Right now, I just kind of want to kind of go through so you have a sense of what's going on here. Corneal reflex. This is basically your cornea is the kind of the clear window into your eyeball. And if you touch somebody's cornea, what do you think the response is going to be?
A punch?
Oh, no, that's kind of secondary. That might be.
I mean, you move the eyelids or close the eye.
Yeah, exactly. You're going to close your eye. You're going to blink. Right. And why does that make sense?
To protect it?
Yeah, exactly. Right. This is an innate reflex designed in your nervous system. If something touches the front of your eyeball, you shut your eyes to protect. So this is something you can test. You don't have to actually poke someone's eyes out. You could just use a little wisp of cotton is very sensitive. You just touch it with a little piece of cotton or thread and it's still gonna trigger it. So, there's no danger of, poking somebody's eye out with a wisp of cotton. So it's a safe way to test a reflex. So we'll be looking again at videos of doing this. You can do this at home. If you do this at home, be gentle. You don't want to accidentally hurt somebody's eye. Pupillary reflex. These are kind of classics. This is where you shine a light in somebody's eye. And what's going to happen to the pupil, the size of your pupil, when you shine a light in somebody's eye?
Constrict, get smaller.
It's going to constrict. It gets smaller. It kind of makes it harder for the light to come in. When you're doing this one, it's important that you pay attention not just to what happens in the eye that you're shining the light in, but that you're at the same time, you're looking at what happens to the pupil that does not have the light in it. Do you all remember what did we mean? What does ipsilateral mean?
Same side.
Same side. Contralateral was opposite side. And when we said that there was a consensual response, what did that mean? If anybody remembers that.
When the unaffected side has a response as well.
Yeah, so it's like you have a normal ipsilateral response, but then you also have a response on the contralateral side that didn't have the stimulus. So again, it's going to be important when you're doing this pupillary light reflex. Don't just look to see is the eye you're shining the light in have a reflex, but make sure you also notice what's happening in the other. When you're doing this, it can be hard to kind of really notice it. Like I did not find any good videos where you could actually see it well because the contrast of the eye is pretty difficult to. So I actually have a video of a kind of simulation of this. But if you do this at home with somebody, you can see the real thing. But in your write-up, you can just describe what you see in the little video I show you. From what we've been talking about, is that going to be sympathetic or parasympathetic that is being responsible for this reflex? If it's making your pupil constrict.
Sympathetic.
Parasympathetic.
Yeah, remember, sympathetic was like fight or flight. Your eyes are like, whoa, right? Parasympathetic is the constricting your eyes. So sympathetic would be dilating your pupils, making them wider, like the wide, I always think of wide-eyed with fright, whoa, versus parasympathetic, which is the constricting. Ciliospinal reflex. This one is where you actually like pinch or poke right behind the neck near the hairline. This one's really hard to see. I do recommend like you just go in the bathroom, turn the lights on kind of low, because if the lights are too bright, or actually it doesn't even matter. The lights are on, look at your pupil, and then kind of pinch the back of your neck. And you should feel it. You should scratch it or pinch it and see if you see any weird changes in the size of your pupil as you're pinching. This one is tricky to elicit, I will say. What time is it? Equilibrium. So the last one's, so gag reflex, everybody's probably done this. If something's going like getting stuck in your throat, you just start having that gag thing going on. It's basically, you can do it without making somebody barf. If you just kind of like stroke the top back of their palate, it's going to create this kind of thing. The whole top of the palate kind of contracts and pulls back. It's something people check. They'll take like a tongue depressor and stroke there and make sure that that response is happening. You know, you don't want to overdo it or do it when you're really full and about to barf or something, because obviously this is sometimes people like make themselves barf by sticking their finger down their throat, and you know, kind of provoking the gag reflex. The other ones in here are all about equilibrium and balance. You know, and we are going to talk about this in much more detail in lecture in the coming week. But the basic idea, in fact, maybe I should just draw it here. Like, here's your skull. In your skull, you have these little semicircular, I'm drawing them way bigger than they are, but you have these semicircular canals. They're kind of like little donuts filled with fluid. And then you have these little, they're called hair cells. And there's like this little cupula, this little gelatinous curtain here. And this detects bending. And we have this fluid in here. And the basic idea is this fluid has inertia. So when you move your head, let's say you are moving your head this way or this way, let's say you're rotating it, you're nodding up and down, the fluid kind of stays still. But this hair cell is attached to your skull. So if the hair bundle, this hair cell is moving this way, but the fluid doesn't move, which way is the hair cell going to bend? If I am pushing this little thing this way, but the fluid is staying still, what's going to happen? Right.
So the hair is going to go the other way?
Yeah, exactly. So for instance, I can oh, here we go. For instance, all right. So inertia, right? Like like here, I have a cup. If I move this paper slow, the cup is moving. But if this cup has inertia, so if I'm kind of moving this kind of fast, notice the cup didn't move, but this thing moved relative to the cup. Did everyone see that? You know, the air in the room is a fluid, right? Air is just a fluid. And here I have this paper. If I'm moving this paper one way, but the air isn't moving, then the paper ends up bending the opposite way, right? This is what's happening to those little hair bundles inside your inner ear when you are turning your head, nodding, or you actually have 3 of these little semicircular canals that can detect whether you're moving your head in the 3 different axes that it can rotate around in. So you've got this going on, right? That's how my brain knows that my head is doing this rotational acceleration, is those little hair bundles in my inner ear bending relative to the fluid. You know, and there's a few different things we use that for. We can use it to tell how our head's moving. We can use it for gaze. Like, let's say I want to just stare straight ahead. I'm staring straight ahead. Maybe I'll get closer. And now that my head's moving, if I want to keep staring straight, I'm staring straight into the camera. My head's moving this way, my eyes have to move off to the side. If I move my head this way, my eyes have to move to the other side, right? If I want to keep staring straight ahead as my head is rotating, my eyes have to rotate in the opposite direction. So I keep staring exactly at the same place. Does that make sense? So what that means is if you fool your inner ear to make it think it's moving and spinning, then your reflex is to make your eyes move back and forth, even though you're not. So like if I, like the clap, I don't know if you can, yeah, I can spin. I start, what happens if I actually start just spinning around like this? Actually, how come right now I am not feeling dizzy or anything? How come I can just loop around here? What's going on? How come I'm not feeling dizzy or actually not even feeling like I'm moving much at all? What's happening to the fluid in my inner ear right now? Anybody?
It's moving as you spin.
Exactly. At this point, I have got the fluid moving along with my skull at the same time. So that means there's no relative motion against those little hair bundles. So the hair bundles aren't bending, which means that I don't really feel like I'm spinning that much. What happens if I stop all of a sudden?
The fluid is still moving, but you're not.
Exactly. The fluid will keep moving, but my head's still. But then as the fluid is moving and pushing against the hair bundles, my brain will think I'm spinning, even though I'm not spinning. So if I try staring straight ahead, my reflex will think I need to move my eyeballs to keep staring straight ahead because my inner ear says I'm spinning, even though I'm not. So I'm gonna like look. I don't know if you can see. Can you like see my eyes at all?
Yeah.
Yep.
I mean, were they moving at all?
Yes.
So there's this, it's called nystagmus. If you're like spinning really, I can do maybe spin a little faster even. If you really get, I'm gonna go faster and faster and faster. And then, and can people see it? I don't know. You can do it at home too. But the basic idea is that is another reflex people check. They will actually make it go. Actually, you can pour warm water in your ears even. Caloric nystagmus, it's called. And it creates little currents in the fluid. And all of a sudden your eyes start flicking. So if your inner ear is telling you you're spinning, even though you're not spinning, that's called nystagmus. So I'm looking at the time. What I would like people to do for this last piece here, we'll have just like a few minutes with each other. Where is it? So there's also these other things that tell you which way is gravity, the sacculus and the utricle. I would like people to do this right now. So this part A, static equilibrium. This is how well can you just balance when you're just standing still. Static equilibrium may be evaluated by having the subject stand with their feet together and arms outstretched. So you're just standing like this and like this. And seeing, like right now, I'm doing it very well. I'm standing, I'm still, I'm not falling over. What information do you have to let you know if you're standing balanced and to keep a good balance? Some of it is the inner ear. We're going to talk about again in more detail. What other things can help you know whether or not you're falling over? We actually talked with the stretch reflexes, right? Just feeling parts of your muscles extending, like you can feel like if you're starting to move, your body's moving, you've got information coming from your muscles and tendons that things are no longer straight up and down. What else? How else do I know like I am no longer straight up and down?
Couldn't you see like your settings change?
Yeah, exactly. I can actually just get visual information about what is my position relative to my surroundings. So what this little thing, let's go share. Test of equilibrium and spatial orientation. So this is page 79 here. It just says, have your subject stand, feet together and arms outstretched. Observe and record body sway. So you just, record like, how are they doing? Or do they need to do anything to stay standing up? Test them first with eyes open, then with eyes closed. So record what you see with eyes open, then have them shut their eyes and now watch what's happening and write down in your lab book what's happening with that. It says, then have subjects stand on one foot and then the other. And then have them do it on one foot with eyes closed. So you're going to have 4 different things here. You're going to have normal standing with eyes closed. You're going to have one foot with eyes closed. And you're going to describe in those conditions, what do you see? And like for the next, we have like 8 minutes here. I would like you to, I'm going to actually throw you into the little breakout rooms. I do want you to actually, you'll have to be able to stand, if you stand back from your little camera, you can pick somebody in your group to be the subject while the rest of you write stuff down maybe. Again, 1 foot up, well, I'm not going to show you right now because I want you to see what happens. But I think it's actually kind of surprising it's actually kind of fun. So I'm going to have you all do this and then we'll just come back in another few minutes. This shouldn't take you that long. So this is going to be like about 7 minutes we have here. And I just we'll report back and just describe what people noticed. So was that kind of interesting?
Yes, it was.
That was super fun.
Yeah. So is your sense of vision that important in balancing?
Yes.
Yeah, I have to say the first time I did this, I was surprised at how much your sight actually helps you like not fall over. So anyway, there's more. We're out of time here, but so we still have a few more of these little reflex things to go over, but I think officially we are at 12:30. So the lab on Thursday it's just you don't have to have anything ready for it. So don't worry about any kind of pre-lab or anything for Thursday. The only thing for Thursday you got to worry about is the quiz. And otherwise, yeah, officially I am in office hours at this point.